me about how you got into doing your PhD.
So basically, I really for me, disease is what I find interesting. How bad things happen to good people.
You want to save the world?
Yeah, save the world. I feel like it's got a really good positive outcome of actually researching something. And so I also really find species interactions really interesting. So how they evolve over time, and just evolutionary theory generally is quite interesting when there's a lot of dynamics going on. And so the sort of what I was looking at for PhD was something that kind of ticked all of those boxes. And this PhD was being advertised for how we can look at good microbes and how they can protect against bad microbes. And I was thinking, well, what's the effect of that going to be on the actual bad microbe? So I thought that ticked, disease, species interactions, evolution, pretty cool implications. I was like, yeah, do it. And so just designed an experiment with that system in mind, with certain questions to answer. And here I am.
What questions did you want to answer?
So there's 3. I feel like I can't talk about all of them because they're accepted and they're coming out, but they're not out yet.
Because you just submitted your thesis.
Yes,
Yay!
finished. I know, good high five. Yeah. Finally, yes. I'm renowned, pretty bad at high fives.
Okay.
So I can talk about obviously the first one because that's this paper. So basically how the good microbe interacts with the bad microbe and actually made it to be less bad.
So what do we know about, because you were talking about when you started your PhD, it was looking at how good microbes could make bad microbes less nasty.
Yeah.
What did we already know at that point when you were starting out?
People have looked at how bad microbes and pathogens and things like that become pathogenic to start with. So why are they harmful? There's been a lot of literature on how they interact with other microbes in the microbiota, but it's mainly been co-infection, so like how a bad bacteria competes with a bad bacteria can make it more harmful. So I really kind of read all of that literature and just lifted it, sort of assuming that actually, if you've got this good microbe, the interactions could still be quite similar. So they could still compete, they could still do stuff like that, and it just happens to not be harmful, so it's actually beneficial. So I kind of made extensions of the theory over to that different field, and they worked, so that's fine. But not a lot has been actually done. So defensive microbes generally, we've only really looked in depth at them over the last few decades. People have known about them for a long time, but we're realizing how common they are. The host that I study is basically a model organism. So when I say model,
you.
It's like it's like a fake host. So it's kind of a surrogate host. So obviously we can't study humans or a lot of animals because it would be really not very nice and also not ethical.
Let's count how many humans die as a result of this treatment.
Exactly, it would be awful. So I use the C. elegans, the little worm.
And where do you get the worms from?
We actually got them from Bristol.
Why Bristol?
Just where a dude picked them out of the soil. They're called Bristol N2.
Ah.
Yeah. But they have to be genetically homogenous. So I had to sort of, they're hermaphrodites. They don't mate with other individuals. So they mate with themselves.
They fertilise themselves.
Basically. And I had to do this 5 times to make sure they had a homogenous population that was control.
So you inbred them to the point where there was no genetic variation.
So all worms were identical. And I actually did name them after you.
Yeah. This is a little fact. She comes to me in the pub one night. Oh, I've got my worms going now. Was it an all-female line?
Yeah, they're all hermaphrodites.
Yeah. I've called them Sally. Should I be honoured? Should I be
Yeah, you should be. And actually, all of my sort of students in the lab that are now started their PhD, they're all using Sally. So
so
Sally goes on for many years.
My scientific legacy will live on.
Yeah, there's probably a tube somewhere in here with your name on it. So yeah.
So you got these Sally worms.
Yeah.
Where do worms, so you say that the dude got them out of the soil in Bristol?
Yeah, I think so.
Is that,
yeah.
what does C. elegans normally do when they're not
being
crushed up by
because they've been
scientists?
Yeah, they've been researched so much that it's so bizarre that we don't actually know a lot about what they do in real life. But basically, they live in the soil. They love rotting fruit because they obviously eat microbes. So they love anything that's covered in microbes. So you'll find them in rotting apples. You'll also find them in slugs. They tend to hitch rides in slugs.
In slugs or on slugs?
I think in, yeah.
So a slug will just eat
Maybe
them?
on. Let's say on.
I don't mind.
No, I think they're on the slugs. Yeah,
Okay.
Can't really remember. So as I said, not a lot is known. Mainly the fact that they live in apples and stuff. But not a lot is known about their natural microbiota either. But my friend in the lab's starting to look at that, and I know a few people will hopefully find more out. But.
And why did you choose tiny microscopic worms called Sally?
Because they're a really, really good model system. So not only are they transparent, so you actually can see the infection, and they're kind of tiny and manageable. You can have loads of them, and they replicate really quickly. So you can do evolution experiments quite quickly.
How quickly?
Like a couple of days.
Okay.
Yeah. So every 4 days, they have gone a whole generation. So it's quite quick.
That's pretty quick.
Yeah. I mean, they're better than you. And me, obviously.
Yeah.
But I'm saying because they're called Sally.
Oh, okay.
That's just me. I mean, you're really unsuccessful.
What do you mean? It's been 4 days and you already don't have a kid?
Yeah, exactly. So every 4 days, they have gone through a whole generation, and they pump out thousands of offspring. They can have hundreds and hundreds and hundreds of offspring from one worm. So, yeah, they can have 900 offspring in a lifetime. Yeah.
900?
Yeah. So we used Staphylococcus aureus, and that is obviously a pathogen of many organisms like humans.
So that's your bad bacteria?
That's my bad bacteria.
Where does it normally hang out?
In animals, so most mammals. In us, it's probably on our skin, in our nose. Yeah, it can be found in our gut, and lots of different places really. But we used that one in the worms because someone's shown before that it can infect and kill worms. So it's already been studied in the worms. So it's quite a nice sort of model pathogen in a way.
So if you're saying it's on our skin, our nose, and our gut, are we always infected?
Usually,
with Staph aureus?
yeah. And it's only really a problem, that particular bacteria is only really a problem if you're immunocompromised. So you might be old or had antibiotics or ill for some other reason. You might have a prior infection or something like that. Or an open wound. So it tends to happen in hospitals when you have catheters, something like that. Burn. Yeah.
What about the nice bacteria?
So that's also isolated from humans.
So pretty much all humans have both Enterococcus faecalis and Staphylococcus aureus.
Yeah.
So everyone has the good and bad bacteria.
Yeah.
And then it's just the number of them that makes you ill or not.
Yeah, it tends to be. So for example, if MRSA or Staphylococcus starts growing where it shouldn't be, or starts growing too well, or starts going out of control, that's usually when it's a bad thing.
And you've taken these two human bacteria species and put them in a worm.
Yeah.
Are they used to worms?
Not at all. Well, we don't really know. No one's found them in worms before. That's actually a pro for us. That's a really good thing. So what's really useful about studying novel interactions like that, where actually they're not natural, so you put a bad microbe and a good microbe into a novel host, you can see how they evolve. Because if you studied them in a host that they were used to, they probably wouldn't change much because they were already adapted.
They're all settled in.
Yeah, they've made it their home. But if you put them into a novel organism, then they have to change because they're not really used to it. And so actually we can measure that change, and we know then what's actually happening, what direction it went in. So it's quite useful. Yeah, it's a good thing to have it in a novel host. This is the kind of a liquid media that we'd grow all the bacteria up in. It goes really cloudy, so you know when it's contaminated, it's gross. This is what we clean the lab with. You can never have too much cleaning when you work with bacteria because that's pretty gross.
Because you're working with human bacteria.
Yeah,
How do you stop yourself from getting infected?
Well, first of all, we wear gloves, which I'm not wearing right now because I'm just showing you around. But we wear gloves, obviously the lab coat, and we clean everything. Yeah, I'm looking very stylish, obviously. This is what I look like.
Oh, outfit of the day.
Oh,
Give us the outfit of the day.
ta-da! Outfit of the day is closed toe shoe, because that would be unsafe with an open toe. We've just got standard jeans. Nice little black top there, which you can see the top of here. And hair up, because we work in a lab.
Yeah. And the lab coat, who designed the lab coat? What are you wearing?
I'm wearing Unibrand.
Very nice.
Unibrand, yeah.
I did clean the lab coats?
Quite a lot, like maybe every month or so, like, yeah, maybe every week or so. I would have made my own lab coat, but it wouldn't have been fire safe, so, you know. I always wanted to make one of like really jazzy colours and stuff, but I wasn't allowed, so that's fine. Yeah, never mind. This is where we keep all our media and stuff that we need to make things with, and our pipettes and all our microscopes. All our incubators are around there, but yeah. So this is our shaking incubator. When it's on, it shakes like crazy and it's kind of irritating. And then all of our other incubators. We have lots of different temperatures for different things, which is useful. That's at 25, classic. The findings of this paper is really important, actually, because if we were to start using defensive microbes in a real life setting, so for example, protecting plants that like crops against sort of crop diseases, and if we were to use defensive microbes to protect endangered species against pathogens or to protect humans against pathogens, there's quite a lot of applications. If we found out that actually the defensive microbe selected for the pathogen or bad microbe to become worse, so harm the host more, then we don't want to use that. It's going to be awful.
Yeah.
But on the other hand, if we find out that the defensive microbe actually selected for the bad microbe to become less bad, that's great. That's like a win-win. Then not only are you protecting the host in the short term from that pathogen, but you're also protecting the whole population in the long term because the pathogens become less pathogenic, less harmful.
As some of these people will already know, a good bacteria, as in a successful bacteria, doesn't want to kill its host too quickly. It doesn't actually want to harm, say, a human that it's infecting before it has the chance to spread.
So it depends on its strategy, really. If you're anthrax, doesn't matter, right? You survive really well in the environment. So actually, if you pop your host, like that's what anthrax does, it's disgusting. If you kill your host really quickly, it doesn't matter. You can sit in the ground and wait for the next one for ages. But mostly, like microbes that are harmful don't want to kill the host too soon. So there is a selection for sort of an optimum virulence level. It usually depends on how that host lives and how the microbe lives.
And what you're finding is that part of how the microbe lives is what other microbes is it interacting with.
Exactly. The whole sort of social context. So for the social context for the microbe is its microbial neighbours inside. I'm saying gut because that's what we mostly talk about, but it's anywhere, like anything. So for the worm, it's the gut. They interact in the gut.
Why does the good bacteria want to kill the bad bacteria?
In this case, it's as simple as it wants the food. So the good bacteria wants to compete with the bad bacteria for us as a host. It just happens to be protective because it's not harmful. In other cases of defensive microbes, they are actively being selected to protect the host. So these these cases are usually when the microbes actually transmitted via the host's offspring.
So when we think of microbes, when we think of bacteria and infections,
Yeah.
we think of something infecting us, making us ill.
Yeah.
So how can something infect us but not make us ill?
Often pathogens, bad microbes are bad because they grow too much, they produce toxins, they can break stuff down that good microbes don't. And actually good microbes often have a lot of sort of beneficial traits. So they break down food for us, or they produce certain things that we need, and they're quite good. And some actually have multiple roles. Some defend us from pathogens, but also give us some good nutrients or whatever. And some are purely one role. And it's been found quite a lot of microbes. It could be on your skin, in your gut, in your eye, in your mouth. Same with any animal or any organism. Plants have them too, that they can basically protect against bad microbes. They can do this in a number of ways. So they can produce toxins against them or antibiotics, which change over time if they evolve.
How long do you think it's going to be before we start infecting humans with good bacteria to cure disease?
We're kind of already doing it. So people at the moment are already doing fecal transplants. That's basically when you get the poop of a healthy person and you you insert it, the rectum of a non-healthy person, and that actually tends to work. So that's great. So that solves C. difficile infection. Not all of it, but a lot of work is being done at the moment, and it looks promising. But also there's a lot of other promising stuff as well. Not all bacteria. So viruses are really selective, and they're really, really promising in this area. So there are these things called phage, and phage are viruses of bacteria. And
so bacteria can also get ill with viral infection.
Yeah, exactly.
So a bacteria can catch the flu?
Yeah, 100%, but die from it all the time, So these phage, which are viruses of bacteria, we can actually use phage to specifically kill the bacteria that are harming us. So it's like the enemy of my enemy is my friend. So this sort of field is called phage therapy, and actually a lot of countries are using it already, and we're not, which is a problem, because actually these phage will protect you, but they'll also be quite persistent in that protection. So if you apply like a load of phage to a burn wound or something, if you get bacteria in that wound that are growing that become resistant, then those phage will actually evolve on the same time scale as the bacteria. And so you get sort of co-evolutionary arms races, even in the time scale of one person's infection. And so you don't really get resistance. Like you get resistance, but it's solved. The phage solves it. It evolves in response.
Because if you're adding a drug, a chemical, the bacteria only has that drug and nothing else to evolve against.
Yeah, it'll evolve against the drug, and the drug can't change because it's not alive. So then it can't be used anymore, and it's a problem. So actually, phage therapy is a really, really good example where we should be able to use it quite soon. And it's really useful.
Cool.
Yeah, it's great.
In humans, do probiotic yogurts actually work? Are these the sort of defensive microbes we're talking about? Yeah.
The problem with probiotics is that they are maybe not as diverse. The actual yogurts are not as diverse as you'd want them to be. This is because a lot of sort of microbes in your gut taste of poo, and they don't make good yogurts. But there's definitely some evidence of them actually being useful. So they don't all go through and just be expensive poo. They are actually good, I think, especially if you're using antibiotics because you've got an infection. You want to really reintroduce good stuff there. But things like lactobacillus that's found in those yogurts, they produce, a lot of them can produce bacteriocins, which are like little harmful things, let's just say toxins, against pathogenic bacteria. That's quite good.
So what can we do to look after our nice bacteria to help evolve them against our nasty bacteria?
Stop using antibiotics. Yeah, be ideal. When it's not necessary, that would be great. There are also things called prebiotics, which more and more research is looking at the moment, but we don't really focus on a lot in the actual applied real life setting. But they are compounds that we can ingest that actually set up a good environment that favours the good bacteria. So it's not probiotic.
So it's not adding bacteria, but it's nourishing the ones that we have.
Yeah, it's setting up a good environment to favour the good ones. Yeah.
That sounds cool.
Yeah.
But of course, your stuff is all done in worms and not humans.
Yeah. So mine's relevant generally on an evolution sort of general questions to most bacteria. So the mechanism of interaction that I found in my system is relevant when there are sort of similar mechanisms or mechanisms with the same outcome in pretty much every other system. Because we didn't study the explicit interaction with the host, this is purely the interaction between the bacteria. It's actually quite relevant to many systems. It's not limited in a way.
So although you're looking at worms, you're not looking at the worm side of things, just two bacteria that happen to be inside a worm.
Yeah, exactly. And although there are inevitable interactions and they might be different, that's hopefully where more research would sort of come in to see actually does this change in different hosts. Also, does this change when there's a more complex microbiota inside the host as well? Because explicitly we studied just those two bacteria.
What are you up to next in life?
I'm moving to Australia, so
yeah.
Basically, I'm going to work on the same thing, but in non-model systems, so real-life systems. So I'm moving to Australia because there's a lot of work being done there on the use of some like good bacteria, again, that infect mosquitoes that protect them from disease like dengue fever, Zika virus, malaria, chikungunya, West Nile, yellow, blah. And so actually, if you introduce these bacteria into the mosquito population, it will protect us humans from those diseases because it won't transmit it.
So the way to protect humans is to protect the mosquitoes, and the way you protect the mosquitoes is to give them good bacteria.
Yeah.
Cool.
Mosquitoes need good bacteria too.
Yeah, we need to look after our mosquitoes. The mosquitoes don't want to hurt us with all these horrible illnesses.
Yeah, they're not the baddies.
No, the bad bacteria are.
Yeah, the viruses and the bad bacteria. Yeah. Not all viruses, don't put that in, that's misleading. Yeah.
Okay.
Cool.
So yeah, thank you very much and bye.
Bye.